Welcome back to Icicle Land. That strange place where icicles are important. We're going to see the thrilling conclusion of the story of icicle physics today. And it will end on a cliffhanger, okay? And so I'm going to talk today about what we can do beyond just measuring the outside profile and looking at different impurities and all that. And recall that if we add impurities to the water and we grow icicles, they grow ripples. So today we're going to play around with fluorescent impurities. And they're going to tell us a lot of strange things about icicles. Before I talk about that, I was provoked to think about, by Menno about this theory of Grae Worster's. Recall that he considers a pure water icicle theory, and he makes a sort of series of models which have higher and higher advection, and he identifies the most unstable wavelength for different rates of advection. And Menno pointed out to me that all of those predictions scale with this thing called the gravity capillary length, which has got surface tension and gravity and the density in it. And in fact, up to a dimensionless number, this is the wavelength. Multiply that by a dimensionless number, which depends on the Peclet number and other things, should give you the wavelength. And this scales as the surface tension to the 1/3. So we can check that scaling with the limited data that I showed you last time. And my student, Anthony Chen, measured the surface tension effect of TX100, which is the surfactant. And as you add TX100, the surface tension drops. This is the bare water surface tension minus infinity, okay, because there's log scale. And as you keep adding TX100, the surface tension drops until you reach this kind of threshold. And it's believed that what happens there is the molecules stop going to the surface and they go into micelles and they go inside the bulk liquid. And so you can't push the surface tension down any farther than that. And this is a common feature of surfactant physics. So just to be safe, so you'll notice that these graphs don't agree with each other, okay? It's hard to measure the surface tension as a function of the concentration, and various people have tried it for TX100, and they get all these different results. But Anthony Chen got these red dots. So what we did was we chose an operating point of 10 to the minus 3 weight in order to be away from all this stuff and to minimize, to drop the surface tension as far as we possibly could. And we can drop it from 0.70728 down to about 0.031, which is actually more than a factor of 2. So what I said before was at least 50%, but it's actually more like 40% of the surface tension. So does it work? The cliffhanger, okay? I told you last time when we looked at our 5 data points with fluorescein-doped icicles, their average wavelength was 8.6 millimeters, and the grand average of all the other runs is 9.39 millimeters, and that gives us a dimensionless ratio of 0.92. And now the prediction is that that ratio should be the same as the ratio of the two surface tensions to the 1/3. Ready? Okay, so we put in the data with error bars, and out comes 0.75 plus or minus 0.02. Okay, now we have this philosophical problem. Are you happy about this? Okay, it is outside the error bars, I have to say. The two are, not 10 standard deviations apart, but maybe a couple, okay? And so, I would say they're not in sufficient agreement to be convincing, I would say. Of course, many of the assumptions that underlying this theory are just not correct, and I'll show you some more today that aren't correct. So we were just trying to recover the scaling. Okay, so I claim that what we need is a whole lot more data. If we had a whole lot more data, we could constrain this ratio more tightly, and then we'd know something. Right now, we have a provocative but inconclusive result. And I'm hoping that Menno has some more data. Okay, so you give me that data, okay? All right, so what happens if you grow a ripply icicle with salt and you cut it down the middle, okay? So this is an icicle sectioned vertically. And what you see is, what we noticed before is that pure water icicles are kind of transparent and doped icicles are kind of foggy. What is this fog, okay? And what you see when you cut it down the middle is you see the fog is not random, it's organized into these stripe-like things, which we call chevrons. And you also notice that there's some structure at the center of the icicle down the middle, okay? And these foggy things were first seen on a natural icicle by Maeno and McConan et al. And here's a funny story, okay? They wrote a paper in which they grew laboratory icicles with distilled water, no ripples. They measured all kinds of things like drip-offs and stuff. And then at the end of that paper, they had a photograph of a Ripley icicle sectioned down the middle, looked just like this. And they said, it is difficult to grow Ripley icicles in the laboratory. See this figure. And then they talked about the ripples for a while. And I thought that meant they'd grown that one in the lab. But no, if you find an obscure Japanese paper, it has the same picture in it. And then you translate that paper, it turns out that it was found on the side of a building in Hokkaido in 1983 or whatever. Okay, so it is not a laboratory icicle. So they didn't know how to grow laboratory icicles with ripples. They said it was difficult. What they meant was they couldn't do it. Okay, so you should never be too polite, okay? Say what you mean, okay? So anyway, this looks just like their Ripley icicle. And they noticed, as I'm about to tell you, that if you look at the center of the icicle and you zoom in on that little box there, the fogginess is not air bubbles. Air bubbles are only seen at the center on the axis of the icicle. And this is because it's only mode 3 growth, the growth of where the pipe fills at the end there, where air is trapped in the water and comes out as air bubbles. So these are air bubbles, but the rest of these foggy things are not air bubbles. So there they are. It's not a very good picture, but there they are, the air bubbles. So what I will show is that these foggy areas here are small fluid-filled inclusions which contain high concentrations of impurities. And the inclusions are organized into this, what I call a chevron pattern, where the maximum of the inclusions are at the peaks of the ripples. Here's a ripple, and here's the black stuff is pure ice, on no inclusions, and then there's inclusions in the next ripple, and so on. And they line up with the peaks of the ripples. And why they seem to go upward like that, of course, is that the ripples grow upward slowly. So this kind of traces the history of the ripple growth. Okay, so what happens as a function of impurity concentration? Again, these are sodium chloride doped icicles for very small concentrations. For basically zero concentration or distilled water, there's not enough inclusion to see anything, so it just looks clear. But if you start putting in more and more salt, what you see is the inclusions start to appear, the fogginess starts to appear, and somewhere in the sort of magic center region where the ripples are nice, you see nice looking inclusion, nice looking chevrons. And as you keep going, things get worse and worse. And finally here, you don't really see much structure. This is mushy ice. Okay? Now, it's not mushy ice like sea ice. Sea ice is so mushy that it can have porous medium flow inside of it. And you only achieve that at high concentration. This is still orders of magnitude less than seawater. Okay? But this ice is sufficiently mushy that it probably has porous medium flow available in it. But this stuff down here is so solid, and the stuff doesn't talk to each other, so I don't think it's porous in that area. Okay, so we're in a regime where the ice is mushy at the extreme of our concentration, but not at lower concentrations. We still see these foggy inclusions. Okay, so now this is a hard experiment. We want to know whether the ripples we measured with topography match the ripples that we see from the cross-section. So now you have to make an icicle, take pictures of it in rotation, keep track of the rotational position of the icicle, and then section it in such a way that you can line up the history of the topography of the shape versus what you see in the final state in the ripples. And when we do that, you can see these green things are the icicle growing. The red dots are edge-detected minima of the ripples, so automatically detected, no eyeball. And you see that they line up, okay, there's some discrepancy, but they line up with the dark regions between the ripples, and the ripples line up with the maxima of the topography. The chevrons line up with the maxima of the topography. And you can, for instance, get a separate measure of the wavelength from here, as you can get the separate measure from the wavelength from the inclusions, from the fogginess, and you can even correlate the angle that the inclusions make with the measured upward speed of the ripple. So it all hangs together. these are the tracks of ripples moving upward with the inclusions concentrated at the peaks. Okay, we did all this in a Phys Rev paper. Okay, so now the fun thing starts. Now you replace the salt with fluorescent dye, specifically fluorescein, our favorite dye. Okay, and then you section the icicle, and we built a box to hold everything at 0 degrees so we could section it and look at it without it melting too fast. And what you see is the fluorescein in the inclusions will glow green if you're close to 0 degrees, and this is actually quite liquid because we've sliced off the two sides. And so this shows that the inclusions shows that the impurities, in this case dye, are concentrated near the peaks of the ripples. And if you zoom in on this little box here, you see something new, which is that there's a substructure of crescent-shaped clusters. The ripples are not only maxima of the impurity concentration, but they also show a kind of layered structure. Okay? So we call these things crescents, and if you cut the icicle the other way, in this direction, so longitudinally, I guess, then you see these things show up as kind of tree rings, okay? And the tree rings were actually observed a long time ago by Charles Knight, who wrote a bunch of papers about natural icicles back in the old days, back in the 80s. And he attributed those rings to air bubbles, okay? But they're not air bubbles, they're impurities. But if you section an icicle this way, you might think that it grew, in rings like this. But if you section it the other way, you see that the rings have a much more complicated structure in the vertical direction. So if I cut through here, I would see some little round things in there. Okay? Now you can see the inclusions with a microscope, a low-power microscope. So I've circled several large inclusions, and they're around microns, around 10 microns in size. And the bands of inclusion-rich ice are roughly 100 microns apart, or 100 microns thick, okay?
So they look like bubbles?
Mm-hmm. They look like bubbles, that's right. They are not. They are liquid-filled, I claim. And I know that because when you look at the dye glows inside them, okay? Okay, so the next thing you can do is say, well, okay, so obviously the impurity is getting into the ice, and it's ending up in these inclusions. How much ends up in the inclusions? And the answer is, if you take the known concentration of the feed water as a function as you increase the concentration of the impurity, then you can put a little cup under your experiment and measure and collect the runoff water, and you measure its concentration, and you find the fraction of concentration in the feed water that ends up in the ice. And it turns out over a wide range, and there's a lot of scatter, but it turns out to be roughly a third. So when you make an icicle out of unclean water, roughly a third of it ends up inside the ice in little pockets, okay? It's a big fraction. I didn't think it would be that big, but it is. Okay, so now we can try to use the dye to visualize what's going on, okay? And this is where the fun begins, I guess you'd say. Watch what happens. These are basically 1 millimolar and 4 millimolar. So this is a fairly high level of impurity by our standard. This is a kind of medium level. So you see the ripples are there, but they're not huge. Here they're huge, okay? What do you see? Well, you see the surface flow is fluorescing. That's the color of fluorescing dye. But you also see that the background icicle is kind of orange. It turns out that fluorescein is magic, okay? When it's inside of an inclusion at high concentration, it stops fluorescing. It quenches, as the saying goes, and it looks orange. And that's great, because that means we can separate the signal from inclusion-trapped dye and surface flow dye. And what you also see, if you go back, let me just go back here and do this again.
Just an effect of the pressure, I guess?
Well, it's believed to be a concentration and temperature effect. I'll come back to that. But here you see it's very orange, okay? And you're going to see a lot of orange at icicles. So the surface under the fluorescent regions, if you put your hand in the box now and you scratch the icicle, like this, with your fingernail, okay, I admit this is a rather crude measurement, it feels rough under the I'm going to give you the word that I want to use here. What this shows is that the surface flow is nothing like a thin liquid film that covers the entire icicle, which is super cooled, which is exactly the assumption of all the models that I showed last time. Once you do not have axisymmetric thin film flow, there is no reason why the fluid has to be supercooled, and I don't think it is. If you scratch the surface of the ice underneath one of these rivulets, as we call them, it feels rough and kind of mushy. If you scratch the orange ice around it, it feels cold and smooth, okay? So what that means, we think, is that the ice is growing under the rivulets in a dendritic way. And that would make sense because it's trapping these little inclusions, and when we've seen the posters and stuff here, where the inclusions come from is that the dendrites are growing so fast they kind of suck up and get little inclusions behind them, and that makes sense, okay? So what this shows right there, that experiment basically blows away all those models of the ripples because they all make these simplifying assumption of axisymmetry and thin film flow, and it's just not like that, okay? And these things we call rivulets. Okay, so when you look at the fluorescence on a dye-doped icicle, so here's lots of good orange for you and lots of good rivulets. What are you seeing? Well, I am going to be very careful to interpret this, okay? The macroscopic fluorescing signal, the green signal here, only indicates regions of macroscopic amounts of water, okay? Liquid water. Only regions with enough dye concentration and thickness of water will give you a signal. There is a threshold below which, and you're going to see icicles like this, where we can't see the fluorescence because there's not enough dye and maybe not enough water, okay? If the dye concentration exceeds a certain threshold, the fluorescence quenches and it turns orange. And it's believed that quenching is also temperature dependent. If the dye solution is cold enough, it will turn orange. And the reason, apparently, I don't understand this, but the reason is that the fluorescing molecules associate into dimers, and the dimers aren't fluorescent. It's not the only dye that does this, but it magically does this around the temperatures that we're interested in. Okay, and we are not just seeing the surface. The dye that's trapped in the inclusion is orange if it's concentrated enough, and as I will show you in a minute, it can also be fluorescent if it's not concentrated enough. Okay, so we don't see just the surface flow here, we see the interior too. And it seems clear, now this is maybe provocative, but it seems clear that the rivulets that we see on the surface contain most, if not all, of the macroscopic surface fluid. Notice that word macroscopic, okay? Okay, what the dye does not show is it does not show whether the liquid is flowing. And in fact, on heavily doped icicles here, it kind of looks stagnant sometimes. Rivulet implies that it's like a little river, but it ain't like a little river. It's more like a little series of lakes or something, right? It doesn't show whether the surface away from the rivulets, i.e. this orange stuff, is wet. And I know that's a fighting word when it comes to ice, okay? I assume that this ice up here is at least like normal ice. It has a quasi-liquid layer, it probably has a pre-melted film, and I don't know what's going on there. My only claim is that this is liquid water, a macroscopic amount, and this is something else. This is not a macroscopic amount of liquid water. It's also possible, in principle, that the orange areas that you see have a macroscopic amount of water on them, but it's quenched for some reason. I don't think I know the reason, but it's logically possible that it's quenched and we can't see the dye there. And I don't know why that would be. It doesn't show at all where the ice is growing. All we have is the topography of the edge. And that's great for looking at the whole size and shape of the icicle, but it's nowhere near sensitive enough to decide if a little tiny bit of ice has grown under a rivulet in a few seconds, okay? For that, you need a probe which can see small amounts of growth. And we can only see a pixel's worth. And a pixel is great for the overall shape, but it's not nearly enough to tell whether the rivulet. So what I don't know is whether these rivulets are growing faster, growing ice, faster than the surroundings. Okay? So the kicker is if you mix, so this is an icicle where all of the impurities are dye. Okay, so it's got 8.3 millimolars per kilogram of dye, and it's very heavily doped. This one has actually slightly less dye, but I've made up the same amount of molarity of impurities by using a whole bunch of salt and a small amount of dye. Okay, so this is a mixed salt and dye experiment, and it ought to look like this one. And I believe that there are rivulets on this icicle, unless something crazy is going on. And the rivulets depend on what the impurity is, but I don't think that's true. But do you see rivulets there? Oh, you do not, okay? So what are we seeing? What we're seeing is the surface flow is glowing, and so are the inclusions. So everything that's orange in this picture is glowing in that picture. And we can't see anything. If you turn off the flow and put your icicle in the freezer, this one looks all orange. And this one glows happily just like that, okay? Because the inclusions are just holding the dye, and there's not enough concentration in the inclusions to quench the fluorescence. That's what we think is happening. Now, the thing we didn't do, which I was urging some people to do, is to reverse engineer the concentration and temperature conditions for which the dye is quenched. And for good measure, for extra marks, you should also measure the concentration of the dye in the inclusions and make all this quantitative. But we didn't do that. All we did was make dyed icicles with no salt in them. We only did a few runs like this. And we interpret the green color to mean liquid water is present on the surface at macroscopic quantities. Whatever else is happening, we don't know, okay? So that's a caveat or a hole. All right, so to analyze the area of the icicle covered by rivulets, we use a trick called false color. This is equivalent to putting a filter on your camera and only seeing the green color from the dye. It turns out the orange and green colors are cleanly separated electronically, if you like, if you project the RGB color space of the photograph into something called the CIE lab color space. And it's just a different way of presenting color, okay? And when you do that, there's a peak of green and a peak of orange, and you can turn all the orange pixels off, and you just see the green ones, okay? So this is the same icicle where you see some orange and some green, and then when you do the filtering, you see just the green and the sort of black and white. Okay, so it's easy to count the pixels here that are green. And then you rotate the icicle 1/16 of a rotation, and you do it again. And you do a clever geometrical trick to unroll the icicle. So you kind of take the icicle and slice it down and do this with it, okay? And we can do all that in software. And you can count how many pixels are above a threshold of the green color. And then you can average that over a rotation. And that gives you the fractional coverage of the icicle by the liquid water that is glowing green. Okay, does that make sense? This is why you hire students who are really good at Python, okay? Okay, so what do we see? Oh, okay, we see that the slide changed when we didn't want it to. Okay, right, so this is a movie with a sliding box, okay? And the sliding box is focusing on the tip. And here's the tip. What you see is perhaps not surprising, but the tip is always wet, always covered with liquid water. And all the drip-off water is fluorescing. But at first, let me wait for the movie to start again here. At first, the top of the icicle is completely green. Wait for it. Come on. Anyway, what happens is the top of the icicle looks completely covered with liquid water. And as it grows, it begins to look a little dry, okay? And by the way, this icicle, these are millimeters. So this icicle is about 50 millimeters long, 50 centimeters long. So here it looks like it's completely covered. But as time goes on, it gets less than completely covered. And when it gets really long, you see the top of this part, the shank, as we would call it, becomes just some rivulets and not much dye coverage. Okay, how does this look as a function of business? Okay, so for high molarity, the icicle looks completely covered for a while. And this is the one, I think this is the same one we just looked at. And as you decrease the amount of dye, and they stop when they've reached their maximum length. And over here is an almost pure one. And if you wait for the movie to start again, which I can do like this, you will see that really you only see the dye at the tip. Now, dyed water is running down this icicle to get to the tip. Do we see it? We do not, okay? So that means that there is at least some rivulets or something where we cannot see enough signal in the dye. Because there's just not much dye there, you know, because I've only put a little tiny bit in. And it's 100% dye, the impurity in this case. Okay, so what we see generically is at first, for the first hour or two, the surface liquid covers the whole icicle, and you get a coverage. This is the coverage over here, liquid fraction. The area of coverage is basically 100%, okay? And then it reaches this kind of threshold where the rivulets start to appear, and you see them, and then the area covered by dye, by liquid water, becomes approximately constant. And the area of the icicle, of course, keeps increasing because it gets bigger and bigger. So this is a bit like cessation. You remember cessation occurred because the area of the icicle just gets bigger and bigger, and the amount of water you're putting in is constant. So eventually those things have to cross over, and it will stop growing when the water gets eaten up before it reaches the tip. Here, it's continuing to grow, but the dyed water that you're putting in at the top is not enough to cover the whole icicle when it gets big. Okay, does that make sense? That's what you expect. Okay, so now Menno did an experiment with dye, and he did something we didn't do, which he used this incredibly pure milli-Q water, which is 10 times more pure than our water, okay? And you may recall that this caused the icicle to become non-platonic in that low limit, okay? In our distilled water, we start to see a kind of platonic icicle, and it's platonic for a while until the impurity makes it not. And he also saw, at that level, he also saw that the tip remains covered with water. This is methyl blue. Now, this is such a small amount of methyl blue that it doesn't constitute an impurity. It's not enough. It's some, though, right? Okay, but he also sees these little drips running down, and we don't see those because the contact angles, in the purest case, are not big enough for us to see them. And then at the opposite extreme, he grew icicles for an hour with methyl blue at about 4 times the highest impurity level that we looked at. And I'm surprised by this because I would expect these ripples to be enormous, but he only grew them for an hour, so they didn't have time to get huge. And this vertical height is, I don't know what it is. What is it? 25 centimeters. Okay, so our icicles are roughly twice as big, and they grow for 4 hours or something. And so had he grown it for 4 hours, oh, and the methyl blue appears to cover the whole surface here. This is completely consistent with what I just told you. If you had grown this thing, 2 or 3 times as long, then it's inevitable, and the flow rate matters too, and then inevitably you would have seen the rivulets eventually. Okay, but the important thing is you were able to measure the contact angle for impure water sitting on impure ice. And our operating point is, this is freezing over here, and everybody's interested in freezing, but we're interested in -13, which is way down here, okay? So at our operating point, as you increase the concentration of dye or any impurity, the contact angle drops rapidly, okay? So this makes sense. As we go to higher and higher impurity concentration, the rivulets get wider and wider, and they cover a larger and larger area of the thing because the contact angle is dropping, and it's wetting more of the ice. Does that make sense? The liquid spreads over a larger area, eventually forming rivulets, and it eventually forms a constant total area of coverage because we're putting in a finite amount of liquid per second, and that just accounts for how much area of rivulets you can get. Okay, so it all kind of hangs together. So here's the area in square millimeters, and there's more details here. We all grow them all for a certain length of time and all that. And as you increase the quantity of dye, the area goes up roughly like a power law. And I don't think this is an important power law, but anyway, that's a way of putting a line through it. And this is a log-log plot. And I think these are different impurities, if I'm not mistaken. No, they're all fluorescent. And I don't remember what the difference between the series is. It might be flow rate and stuff, okay? So the area covered by the liquids on the surface increases rapidly with concentration, as I showed you in the picture. Another thing you can do is you can look, where do the rivulets go? And it's interesting to ask, what is the correlation between the places that are wet with fluorescent dye and the ripples? This is what we're trying to understand. So if you look at a picture like this is a fairly heavily doped icicle. It's filtered, orange filtered, so there's no orange in there. And what you'll sort of notice with your eye is that the green stuff seems to be on the top side of the ripples. And indeed, this is the signal. This is the right-hand edge. Then we rotate the icicle 45 degrees and take a strip down the middle and measure the dye there, okay? And that's the so-called liquid, the orange stuff. I should have made the line green now that I think of it, okay? And you can do a cross-correlation between those two signals, and you find a peak. And the correlation is peaked at about 1/3 of a wavelength. So that means the dye, all things being equal, is a third of the wavelength up from the peaks of the ripples, right? So it's on the top side preferentially. Now that makes sense because fluid mechanically, as you're flowing down, you're going to linger on the topography, and then you're going to go fast on the bottom. It also helps explain, and if let's say, the ripples are growing fast where the liquid water is, that would explain why the ripples appear to move upward. The ripples don't move. What happens is the ice grows on the top side preferentially, roughly a third of a wavelength up, and that gives you the stacking, that gives you the chevrons, and also gives you the rising ripples, okay? So it all kind of hangs together. Okay, so we should have stopped there, probably. But I decided that what we wanted to do is separate the shape problem from the ripple problem. And one way to do that is to take a page from Matsuda, who you remember wrote a thesis which was never published, where he measured ripples for tap water on the sides of a stick. So I call this ice on a stick. It's like a, a ice lolly experiment, okay? And the stick is wettable wood. And so there's no problem getting water to cover the stick. And sure enough, it grows nice sample ripples, which are all the same up vertically. So I was able to do this in the old Cambridge experiment. I used a wooden stick, and I can see beautiful ripples growing on the side of the stick. So this is an experiment we wanted to try. So what we did was, oh, and by the way, this does happen in nature. My neighbor in Toronto has a really bad drainage system on his roof, okay? And this is a steel drain pipe 4 inches in diameter, okay, about that big. And water does not go down through the pipe. It runs over the surface of the pipe, and it's rough. And so it does the experiment for me every winter, okay? And so this wavelength is a centimeter, and you can see how it's much bigger than the pipe. So somebody asked me, what happens if are the ripples correlated around the pipe? And the answer is, for small icicles, they are. And if you were somehow to make an icicle 4 inches in diameter, which I have done, then the ripples lose their coherence around the outside. And that is what you get, ripply junk, okay? So this is like a crossover between flowstone and stalactite or something like that. Okay, so what we're going to do is we're going to do a cylinder experiment. We're going to start with a preformed cylinder made of distilled water ice. And it's hard to make, so you put a string down the middle so it doesn't fall apart, okay? Then you flow fluorescein-doped water over it at a concentration where we expect to see ripples. And we expect to see the ripples appropriate to that concentration. That's what I believe naively would happen. Do you believe that? How many people think I should see beautiful ripples in this experiment? Okay, put your money down, okay? What happens is, it's rotating. You can see the dye flowing down the outside. I'll show you a visualization of that in a second. You can see the cylinder getting bigger. And in fact, it grows by a factor of 3. It gets 3 times the diameter it did at the beginning of the experiment. What are you not seeing? You are not seeing ripples, or at least not very big ones. So the puzzling result is that the impure ice thickens the cylinder okay, but no ripples seem to form, even though I expect them. So I naively thought that we were doing a stick experiment to study the ripples without the difficulty of the shape of and the growth of the icicle, but it fooled us, okay? It did something new. Okay, so here's what we see. The puzzling result is the impure ice forms and the cylinder gets thicker, and you can even see, if you filter, you can see the rivulets running down the outside, and you can see the orange inside the ice here and there. So things are happening, but nothing is in the ripple height, okay? So we observe rivulets and some dye trapped in the ice, but it does not behave like an icicle made with the same concentration. And just to show you that's true, here's an icicle with actually a smaller concentration than the cylinder. And that's the kind of ripples I would have expected to appear. Now, this is made with salt, okay? But we believe that this is a colligative problem, and so it doesn't matter what the species is. But here I claim that these ripples are much bigger than you see on this dye, on this experiment, right? Okay, so now we get desperate. What do? We take, this is the original experiment, we take the experiment and we carve grooves in it. Let me just go back. So here's a groove, and it's a deep groove, okay? It's being held together by string. And there's actually some smaller grooves, you've probably lost them here. There's one down there. And then we do the experiment again. Now what do you see? You see a heck of a big ripple right there. And you even see some above it, okay? And you see a little bit of ripples down here. And you see the orange is much more prominent in here than it was before. So now we see some ripples form, mostly above the deep groove. And we observe more trapped dye than we did in the first experiment. And these are exactly the same conditions, only the topography is different. Okay, so here's a fun experiment. You make a cylinder of ice, you put it in the icicle machine, and it breaks, okay? The thing fell off. So we did an experiment with a truncated cylinder, which we didn't intend to do. And this is like it's mocking us, right? We have a cylinder experiment up here, and we have an icicle experiment down here. And what do you see? Well, the icicle looks way different than the cylinder. Okay, so we start with a truncated preformed cylinder made of distilled water ice, and now the fluorescent doped water does just what it did before, below, but at the top it does what it did in the previous experiment. It doesn't grow any ripples. And even more insulting, there's a ripple that grows upward from the junction here. Okay, so the interesting result is that the cylinder thickens, but only kind of slowly. This is thicker than the cylinder. And a typical icicle, ripply icicle, grows before. And one ripple grows above the break point. So what these experiments, to me, suggest is that the internal structure of the ice has to be, yeah?
If you change the initial diameter of cylinder, do you still, and cut it, for instance, do you still have this separation of cylinder and icicle?
We didn't do too many experiments, so I can't say what would happen if we, I expect that this effect will occur for all reasonable diameters. But I haven't done the experiment. Okay, so what I think this means is that when you're going to form ripples, the substrate over which you are flowing has to be right, okay? It probably helps if the substrate is previously formed ice that's of the same concentration. If we start on pure water, which we did here, it may be that it just never gets going, never starts, okay? Second thing that this tells me is that topography might be important. That all icicles, even distilled waters, are bumpy, okay? And when we saw the ripples on the lightly doped icicles, we noticed that they don't cover the whole icicle. They're kind of patchy. They appear and disappear, and the ripples move upward. This is all what you expect for a convective instability. That is to say, if you make a perturbation, a finite perturbation to launch a ripple, it will launch a patch which then moves upward. It could either move upward or downward, and those are both convective. But because there's no symmetry in this problem, the water runs down. That means that the ripples generically will move, and we know that they move upward mostly, right? So what I think is happening is that we are triggering a low threshold non-linear instability, okay? Not a thing you can reach with linear stability analysis, where the substrate structure has to be right to get that trigger to go. And once it goes, then you get a convectively unstable system, and the ripples grow and they move upward. Okay? That's a picture which is consistent with what I have just shown you. Yeah?
Do you try to run ice in a steep while varying quite rapidly the flow rate?
No.
But I guess
Life is short, okay? I first thought, oh, let's just steadily increase the flow rate so that we don't have this problem of running out of water. But, you know, these are, like, each of these experiments takes a day. So we haven't done a lot of things.
you could argue that the development of instabilities comes down to geometrical imperfections that will impact locally the flow rate.
Okay, I'll tell you where I think the imperfections, they're not imperfections, they're perturbations of the smooth surface. I would suggest that it is not the case that ripples must start near a narrow tip, because I just showed you a drain pipe which is 4 inches in diameter covered with ripples, and there's no tip there. Also, you can do flowstone-like experiments where there's no icicle at all, and they get ripples there too. I think the ripples are triggered by, well, I'll tell you. I'll show you what I think they're triggered by, okay? But the point is, this is another blow to linear stability analysis, because linear stability analysis cannot reach finite amplitude problems like this. Yes?
Were you saying that the lack of impurities in the initial cylinder were causing this effect? Like, what if you had impurities in your cylinder?
Sadly, we did not do that experiment. But I suspect that when the ripple is deciding whether to grow or not, it's happier, if you allow me to anthropomorphize, if the ice over which it is flowing has a pattern of impurities in it from the previous flow, and off it goes. It's happy then, okay? If it flows over pure water, then it behaves as if the impurities don't matter, and it behaves like pure water. But the real point, from the point of view of analysis, is that this is a finite amplitude instability. This is what it looks like. I was only able to get ripples on that cylinder by sawing grooves in it, okay? Okay, so if you're doing linear stability analysis, I'm sorry, because the base state that you are assuming is not axisymmetric, so your entire theory is nonsense. And worse yet, linear stability can't reach a finite amplitude. And also, if it's convective, you need a different analysis. Sorry, okay? All right, so what are the perturbations? What could be the thing that causes the icicle to be bumpy? The answer is, of course, that icicles are like snowflakes. No two icicles are exactly the same. The reason they're like that is that they are not homogeneous solids. They are polycrystalline. And these are not my pictures. This is Matsuda again. He sectioned to some icicles. And there's a beautiful paper by Gerald Walker, an amateur scientist. He talked to the guy, Mr. Knight, and they wrote a little article about all the beautiful things about the stuff. And he produced this beautiful picture, which, as far as I know, not published anywhere else. And so these are the crystallites. Okay, and this is a transverse section, and the previous one was a longitudinal section. So this is the vertical axis here. So we did that, and the reason we did that was we had Ripley icicles. We wanted to see what the correlation between the ripples and the crystal structure was. And the answer is there is some. So here is a vertical section, and what you notice, first of all, is this is what it looks like in ordinary light. You see the mode 3 growth and the bubbles. And here you see that's a single crystal, big, long single crystal. And so when mode 3 grows at the tip, it tends, not perfectly, but it tends to produce a single crystal oriented that way. Okay, and you can see that even more in this picture here. This is a single crystal oriented down here. Now, if you look here, admittedly, this is purely eyeballing, okay? There's no analysis here. But here you see crystallites which are sort of trending upward. And those are correlated with the ripples. So I think what's happening is we're making an icicle which has its own internal crystal structure, and it's bumpy because of crystal structure. And remember, when we turned the air fans off, the thing grew legs and dripped from other places, okay? So that's also due to crystal. So you can't do the same icicle experiment twice because the crystal structure is different between the two. And even a pure water, even the most platonic pure water icicle, is a little bit bumpy. And the reason it's a little bit bumpy, as we all know, is that the crystal structure, the crystals grow a little faster, a little slower in different directions. And that means that the bumps are kind of inevitable. So it's kind of an intrinsically noisy instability if there is an instability. So these bumps trigger a finite amplitude instability, convective. That would be my picture. And some grains actually span several ripples. So they're not perfectly correlated. They're very slightly correlated with each other. And I have to say, these type of crystal stuff was already published by Knight in the same paper as I showed you the cross-section. He also has pictures of the cross-section. Okay, now these are difficult to interpret. I'm not an expert on crystallography, and so all we did was eyeball this, okay? Okay, we have reached the thrilling conclusion, which is, for you students, homework. Here's my list of homework questions. Perhaps you can think of other ones. We measured the temperature of the air. What we did not do is measure the temperature of a rivulet or of the surface of the ice between the rivulets or what was going on at the tip. You might say, oh, the tip must be colder than the top. Well, I don't know whether it's colder than the top. I can't measure it. And I tried an infrared camera once, but it didn't have enough resolution to see the differences in temperature. Maybe you have one. I mentioned this already. We never reverse engineered the dye or tried different dyes or tried different concentrations of dye and salt. And there's a whole spectrum of possibilities there, which I would like someone to do. The surface tension, we did 5 experiments where we dropped the surface tension with the maximum amount, but we didn't do it as a function of surface tension. Maybe Menno has some data there, but that's a thing we should do. And I saw a poster about inclusion formation. I don't understand how the inclusions form. What determines the density and composition of them? If I was to make a linear stability model where I had a boundary condition on the concentration at the growing surface, I need to know how big the flux of that thing is into the ice. John Layden and I just assumed it was 0 in that model that I showed you. But it's not 0. How big is it? I don't know. I haven't got any model. Okay. Ripples, are they really a linear instability, or are they punishing us by being nonlinear? Why are the impurities really required? One idea is simply that the impurities help you cover the whole surface of the ice, and then Bob's your uncle, the impurities don't matter, okay? But I suspect that the three facts, the ripple topography, the inclusions, and the third thing. Anyway. Oh, the fact that you need impurities. Those are all coupled with each other. You don't get inclusions without impurities. You don't get ripples without inclusions. You don't get inclusions without, you can't make a model which leaves out one of those things, okay? Grae Worster and Wayno made pure fluid model, pure water models, and I don't think you're going to get the right impurity. You're not going to get the instability there. And it's not just not just because the surface is covered when you add impurities. The impurities do things, and they're needed to make the thing happen. And I have to say that we are not sensitive enough to see where the ice is actually growing. It is conceivable that it grows under the ripples fast and slow elsewhere. Like there could be, God forbid, a pre-melted film, which is somehow fed by the ripples, and there's ice growing outside of the ripples, outside of the rivulets, I mean. Okay, we can't see it. We haven't got the resolution to see it. And so what I would like someone to do is a single rivulet experiment with enough resolution with dye or with impurities to see where it's growing. And there is a paper by Monnier et al. Who did a beautiful single rivulet experiment, sadly without impurities. And then if you're still interested in macroscopic shape after all this, I complained when I showed you the Platonic icicle theory that we never did anything with the tip. Obviously, as we learned from Anne Montgrel's beautiful experiments, there's lots of interesting stuff happening at the tip. Somebody has to figure out the tip. So you could do an icicle experiment and just forget the rest of the icicle and just tell me what's going on at the tip. And mode 3 growth, and how long is that liquid tube, and all those things. We did nothing about any of that, okay? And the theory just throws it away. Okay, and there's lots of physics going on at the tip. Nobody's studied any of it. Okay, so I wish to conclude by saying that I didn't do any of this, okay? I get to go to beautiful Corsica and talk about it, but the young men who did that are John Layden and Anthony Chen, who are both gainfully employed as we speak. And this is us on the day we knocked down the icicle machine. We took it down and we put it in the museum. So if you visit the University of Toronto, I'm not physically present there anymore, but if you go there and you ask somebody, can I see the icicle growing machine? I'm a historian. They will take you to something called the University of Toronto Scientific Instrument Collection, and we have 125 years of weird old scientific instruments in the basement. Okay, and now the icicle machine has also retired, and it is down in the basement. Okay, and this is actually the first version, and this is like the final version. Okay, well, thanks for your attention, everybody.